Hello my friends, welcome to a video on the somatic sensations. We just finished kind of an overview of sensation and perception and receptors. We're going to dive in deep now. This is it's not an overview, it is deep into the somatic receptors of the integumentary system and the proprioceptor receptors of the muscles in the joints. So it's going to be general somatic, no special. We're leaving that off for chapter 17. And no visceral in this particular video. We'll leave that. We kind of already done that in the autonomic nervous system. So this will be a deep, in-depth view of the receptors of the integumentary system. We met them all in chapter 5, and we talked briefly about them and in the integumentary system. But now we're really going to get into a little bit more detail. Merkel's discs, Meissner's corpuscles, remember Pacinian, hair root plexuses, all those on the left, as well as the three proprioceptive, joint kinesthetic and muscle spindles and tendon organs. I'll take you in a little bit more detail about those. In terms of A through H, what type of sensations they pick up, a lot of overlap too. Quite a bit of overlap that we're going to see. You'll see something like Meissner's is picking up pressure, touch, and vibration. Okay, and then we'll talk about whether they're slow or fast adapting. And there's it's a little sinuous there on the slow or fast adapting. There's a lot of subtlety and a lot of kind of bends in the road, but we'll review it. We won't stress too much about it, but just maybe try to give you a couple key ideas as to associating or categorizing slow and fast receptors. Which of these adapt slowly and which of them adapt really fast. Okay, 4 types of sensation: tactile, thermal, thermal, let me try thermal there, pain, and proprioceptive are the 4 types of sensations that you're going to want. And this is like the 6th different way that we've categorized sensation, but in terms of looking at all the different general somatic sensations that you can feel, not many of these are visceral at all. These are general somatic in your integumentary system, in your muscles, and that you can feel are going to be tactile, thermal, pain, or proprioceptive. So we'll start with tactile, physical sensations: itch, tickle, touch, pressure, vibration. Itch and tickle are covered, and remember this diagram from the last. If you're unsure what this diagram is all about, go back to the last module. Notice we took off the special. Down at the bottom was the 3rd special, right? Sight, taste, hearing, smell. That. Not looking at those right now. We're looking at the general somatic, and itch and tickle are handled by free nerve endings, just sticking out into the dermis and the epidermis of the integumentary system. And touch, pressure, and vibration are handled by capsulated fibers. So those encapsulated nerve endings. So those nerve endings now wrapped in a connective tissue capsule. So when the connective tissue capsule gets pushed or bent or under some physical force, then it moves the nerve endings inside and opens up channels. Okay? So that's just a way for you to separate those out. And then let's get into a little bit more detail about which specific receptors. So here are your 6 receptors in the integumentary system that you can kind of count on. The free nerve endings, and they're in the epidermis and dermis. And also don't be confused. There's also free nerve endings that just stick right into the dermis too, right? Just random free nerve endings. And then your Merkel's, which are these cells that sit right on top. They sit in the stratum basale, but they sit right on top of receptors that are in the dermis. And then Meissner's, Ruffini, sort of specialized encapsulated corpuscle here. The hair root plexus, or the paratracheal, and then the good old-fashioned lamellated or Pacinian, very, very deep. And I know we looked at Meissner's and Pacinian in a lot of detail in 40A, so these will just be refreshed on that. But let's start picking up where each one of these is. Each of these 5 tactile sensations, you can see the overlap. Touch is picked up at 4 different places, okay? Fine touch, obviously, hopefully not too confusing, is picked up superficially, right? Very light, we call it discriminative touch. You can feel maybe a pencil scraping on you versus the eraser end, based on the pencil is sharper and a smaller area versus an eraser lightly pushing against you, right? Discriminative touch. Obviously picked up superficially, right? So the free nerve endings that are sitting superficially, and the Meissner's corpuscles, which are right in those dermal papillae, right? These little infoldings, these dermal papillae, those Meissner's corpuscles are great for just tucking right up under there. So the slightest downward pressure, discriminative touch on the stratum corneum will be relayed through and picked up by both the free nerve endings and the Meissner's. Whereas the crude touch can be picked up deeper, right? The hair root, the paratracheal, pushing down hard, pushing on those hairs, you'll pick that up, as well as just the pressure on your integumentary system, pushing with crude touch is going to be picked up deeper in the lamellated corpuscles. Makes sense, right? Light touch, superficial, heavier touch, done deep, picked up deeper. What about pressure? 3 different places, okay? So again, superficially, picking up light pressure in the same two, the Merkel's discs and the Meissner's corpuscles, and then the Pacinian corpuscle deep for heavier pressure, right? Sort of more full or deeper pressure. So same concept. Light pressure superficially, heavier pressure deep. Piece of cake. We're flying. Vibration. Slower vibration, going to be picked up by Meissner superficially. Fast vibration is going to be picked up deeper in the lamellated corpuscle. Again, fairly straightforward. And that leaves us itch and tickle, and those are going to be handled by the free nerve endings, either epidermally, or of course it could be some, I'll continue to draw this every time I erase it. It's kind of fun to draw. Even some pure dermal nerve, free nerve endings too, right? And these are free, these would be free, but they have Merkel's discs on top. But this also gives you the idea of free nerve endings too. And those free nerve endings are picking up itch and tickle. So those are your 5 tactile sensations and how they're spread around these 6 different types of receptors. Let's move to thermal, right? Here you're going to have special proteins in the free nerve endings. Thermal's picked up by free nerve endings. And just go back and compare. The thermal is only by free nerve endings. What were your tactile? Well, sure, some were free nerve endings, but others were lamellated corpuscles or encapsulated corpuscles. But thermal is only free nerve endings, just the dendrites. Okay? And there's special proteins in there that expand in heat and contract in cold, like everything else in the world expands when it gets warm and contracts when it gets cold. And that's going to be a case of free nerve endings, and those proteins in there, special integral membrane proteins, will change shape, and when they do, they will allow for transduction, opening and closing channels to sense really cold and really hot. All right. So the warm is handled a little deeper in the dermis, and the cold is handled a little more superficially in the epidermis, the stratum basale. Okay. And generally, body temperature is sitting at 37, but anywhere from low 30s is really typical. Your fingers and extremities tend to be in the low 30s Celsius. Anywhere from, say, 32, 33 to 40 is hard to pick up. Like, it's very difficult to pick up. But from 10 to 32, you're definitely picking up like, ooh, that's cold, right? And from 40 to 48, you're picking up like, ooh, that's hot, okay? But lower than 10, that's not cold, that's painful. Higher than 48, that's scaldingly painful, right? That's not high temperature, that's picked up as pain. So, for whatever that's worth. And then speaking of pain, so important, right? And nociceptors are the special receptors in the free nerve endings, picked up in the free nerve endings here. And they just adapt so slowly because pain is just so frustrating. Chronic pain is so frustrating, but it's so important. It's so important. If chronic pain gets you into the doctor to have something checked out that could save your life, that's critical, right? So it's just, they adapt so slowly and it can be so frustrating. And even after the physical stimulus is gone, the pain remains, right? Chemicals, bradykinin, prostaglandins that you continue to secrete are your internal, sort of your endogenous pain signals, as well as any sort of introduced toxin, bee venom being the one that we constantly use as going right in your skin and eliciting, ow, right? Intense thermal, I talk about super low, you snowball fight with no gloves, and that stabbing pain in your hand, right? Mechanical stimulation, i.e. crush, bruising, smashing your skin, wrenching, twisting, and then chemical, right? Chemical. So again, the bee venom or acid spilled on your skin, something like that, where it really just kind of chemically burns you or chemically produces pain signals. Okay. Fast is external. It's really superficial, like ow, you got poked, you looked right at the spot. Slow, more visceral, right? Kind of burning, aching, kind of deeper, more visceral feeling. Or could be just more deep in terms of in the muscles or even in the joints, could be deeper, sort of arthritic, slower burning, aching pain. And there's between the fast and the sharp and the slow. We all know this. You also know if you're in earthquake country with us here, you know, you get somebody who's been through a lot of earthquakes and that first little hard, quick, fast, explosive shake, that's not what you're thinking. You don't give about that at all. You're waiting for that to end to see if it was a 3 or 4.0 with the shaking, or if that fast is over and you wait for that slow, unbelievably powerful wave to pick you up and hurl you through space, if it's a big boy earthquake, right? And same concept with the slow and the fast pain. You stub your toe, instantly there's pain, and then what do? Well, first thing you do is you look around, make sure nobody saw, right? Make sure your integrity is intact, right? Your shame. Second thing you do is you wait for that slower, how bad is it going to be, right? You know, bam, oh, stub your toe. But then you're waiting, right? You're waiting for it to come up on the slow path. Like, how painful is this really going to be? Is this just a quick one and done, or did I maybe even break or fracture something? Or, you know, you're waiting in trepidation for that slow pain to come up. And obviously a lot more chronic and a lot more powerful than the short, fast pain. And then in terms of localization or finding it, fast, superficial pain is not a problem at all. You look right where you got to know. You immediately just look, ow, right? You look right at the spot that you just suffered an injury. Slow somatic pain is also very well localized, and that's going to be, you know, a bruise that happened a week ago and it's still kind of harming, or a joint that's just kind of sore, or muscle pain that's kind of still there from maybe a workout 4 or 5 days ago. It's pretty easy. Your triceps are hurting, right? Your hammies, you localize that pretty easily. It's the slow visceral pain that can be a little sometimes tough to localize because it can be referred to other areas. And these maps here are really helpful, and this is something in a clinical application you'd really want to be able to get your head around, these localizations right here. Okay, you're in a clinical setting and someone comes in complaining of pain in the sacral area, right around the sacrum. Okay, first thing you ask, well, did you fall on your sacrum or your tailbone? No, I haven't fallen in forever. Okay, did you kind of get whacked or bumped or anything there? No, not at all. Okay, then you do maybe a topical, an integumentary examination. No rash, no bump, no redness. Okay, now you're starting to trigger back of your head. Maybe that's a problem with the urinary bladder being referred to the sacral region, right? So then you're starting to talk about the referred pain. And of course the heart, like you get these areas right here, right? Someone's having a heart attack or some kind of heart infarction, and their left arm, you're looking to see if their left arm is tingling or numb or painful. Why? Well, because cardiac pain is referred down to those areas. Anyway, just something to keep in mind in terms of pain. And then finally, proprioceptive. We'll take a little time here just to be sure you guys understand. Muscle spindles are they're really intricate in how they work. I kind of find them fascinating, but they do take some time to get your head around. Don't stress every other muscle fiber in the muscle. All these millions and millions and millions and billions of muscle fibers are all called extrafusal, and we've looked at those in 40A to death. They work on a simple alpha motor neuron. Okay, stimulated, boom, done. We're talking about intrafusal fibers now, and those are very special fibers that are inside of a connective tissue capsule. Here the capsule is shown cut open, but this would all be covered over in gray in this image. Connective tissue. Even the neurons, even the sensory neurons, the afferent neurons are also covered in connective tissue too. And what are these doing? Well, they're measuring and they're just keeping track to the nervous system of your muscle movement. Sure, the alpha motor neuron comes down in response to, let's say, a pain receptor, a nociceptor in the free nerve endings in the dermis is triggered by a bee sting, right to the CNS. Right to an alpha motor neuron, yank your hand away, right? Try to swat that bee away. That's a piece of cake. But then on the back end, you're going to want to record, well, what did the muscle just do? Because you constantly want to know the length of your muscles. You constantly want to know the extent of contraction and relaxation and the length of every single one of your muscles. If you know the length of every one of your muscles and the position of every joint in space, you're going to be in really good shape proprioceptively. Balance and coordination, equilibrium are going to be in really good shape. So these muscle spindles are just simply trying to keep track of how much you're contracting or relaxing a muscle. And you might know these from the reflex arcs, right? The stretch reflex, where you kind of bang the patellar ligament. Boom, you bang that and then you respond to it. You sense the quads being stretched. That's from these muscle spindles. And so you contract in response to it. That's from these muscle spindles. And you can see they're being innervated and sent to the CNS along these blue arrows saying, hey, we just stretched a little bit. Or, hey, we just relaxed. And you want to constantly keep that in check. And you also want the muscle spindles, not only are they sending information, but look at these red arrows coming back. they're being innervated via gamma neurons, not alpha motor neurons, via gamma motor neurons. They're being stimulated to contract just the intrafusal fibers. And what is that all about? You just got this mini little, you got all these extrafusal fibers that are contracting the muscle, and you have this little mini proportional set of interfusal fibers, and they're just contracting to stay in proportion. If the extrafusal are completely relaxed, expect the interfusal to be completely relaxed. If the extrafusal contract just a touch, expect the interfusal to contract just a touch. If the extrafusal contract fully, expect the interfusal to contract fully to be in parallel with them. And why? It's to take the slack out and keep the tone and be in order to be responsive. Okay? So you want these interfusal to be in proportion with the extrafusal so they can respond. So even if you're 99% contracted on your extrafusal and you contract that 100%, you can pick that up if you've got your interfusal also heavily contracted too. All right? A little tricky to get your head around, but let's just take a look at this concept in green. So picture you and a person, you're 5 feet apart and you have 5 feet of rope between the two and you're each holding it. Person A tugs on that rope. What's person B going to notice? Whoa, I just got tugged, and you're going to know immediately, right? Now picture 20 feet of rope between the two, and you're each holding the end, and you're 5 feet apart. What does that mean? Well, you got 15 feet of rope looped on the ground, slack. Person A tugs on the rope. Person B has no clue. Person A continues to tug on the rope. Person B has no clue. The slack is being tugged. So what do? What does an interfusal muscle spindle do? It takes up that slack, pulls up that extra 15 and holds it, so you still got only 5 feet of rope between the two of you. Now every tug is felt. So that's what that taking up that slack is all about, and being able to respond. It's a very proportional sort of scale thing. In other words, if you have a multimeter and you're doing electrical work and you want to pick up a change of 1 or 2 volts in a small electronics device, you don't set the multimeter on 0 to 500 volts because you won't see 1 or 2 volts. It'll come and go. You won't even notice it. You set the multimeter on 5 volts. 0 to 5 is your scale, really sensitive, so that 1 or 2 volts is picked up as moving your needle, so to speak. What if you want to look at your 240 volt circuit on your dryer? You don't set your multimeter on 0 to 20. 240 will just peg the needle and you won't be able to notice anything. You won't notice any change whatsoever. You set it on 0 to 500 so you have a good scale. So you adjust the scale so that you have good response, and that's what the interfusal muscle spindles are doing. Again, I hope that helps. And again, we know these from the stretch reflex. So go back to the beginning of the chapter and the beginning of the quarter and look at some of those reflex arcs and see if you can pick up that stretch reflex where you bang that tendon, quadriceps stretch, these muscle fibers, interfusal muscle spindles send signal, and then you stretch quadriceps, but then you come back, you contract quadriceps in response, but then you come back and then you want to contract the actual interfusal fibers too, the muscle spindle fibers, to keep in proportion with the contraction of the muscle around it. So you're always in tow and you have a good sense of scale. I hope that explanation worked. Tendon organs are a lot simpler than what I just went through. They're in the tendon. They're free nerve endings in the tendon. They're only sensory. There is no response to this. There is no motor coming back. There's no gamma motor coming back. It's simply sensation, and it's sensation to the CNS saying, we are about contracted to the max. We are about to tear. Stop contracting. Boom, signal sent. You know this. You're power curling with the guns, and you got so much weight on there. You're exhausted. You know what's going to happen when you push it. You've seen this, and you just drop the weight. You are completely stimulated to just relax the muscle and drop that weight so you don't tear up your tendons and your ligaments and your muscles. Okay? Little bit simple there. And then finally, your joint kinesthetic receptors. I'll talk about these, how they actually, when we do the muscle spindles and the tendon organs, I'll talk about how slow they adapt, because you really always want to know the length of your muscles, and you always want to know if you're about to tear some muscle badly. But the joint kinesthetic receptors, they're measuring acceleration and deceleration in the joints, and they're going to adapt really, really fast, because once that acceleration and deceleration are gone, you want to just basically ignore it and wait for the next acceleration or deceleration. So a constant acceleration and deceleration of a joint is sort of tuned out pretty fast. It's a little bit different. Anyway, acceleration, deceleration of the joints. Kinesthesia is the perception of body movement. Kinesiology is a study. And I'll just go back to this image again. And this person, this athlete with these external joint kinesthetic receptors, you've got these built into free nerve endings and Ruffini corpuscles, which measure stretch a little bit and pressure inside of your articular capsules, inside of those beautiful joint articular capsules. And they're picking up the acceleration of your elbow laterally or the deceleration of your knee joint vertically. And it's just picking that up in space and giving you that great proprioceptive kinesthetic information that you need. All right, here we go. Let's iron this all out, shall we? Remember, a lot of overlap on these. Quite a bit of overlap. Merkel's discs are going to overlap with pressure and with touch. Pressure and with touch. Remember, those are sitting right on top of the free nerve endings. They're just in the stratum basale. Light touch, light discriminative touch, and light pressure superficially, just discriminative, picking up by Merkel's tactile discs. Joint kinesthetic receptors are obviously proprioceptive. Meissner's are picking up, same thing, light pressure, light touch, and slow vibration. Again, light discriminative, very superficial. Meissner's are superficial. They're tucked in those dermal papilla, so they're as far up in the dermis as you can go and just below the epidermis. Muscle spindles, proprioceptive. We knew that one. Ruffini corpuscles. So these are actually kind of, they do pick up touch, but I'm actually going to put free-forming. Ruffini corpuscles can actually pick up stretch. We talk about stretch as being really important in where? In the viscera, right? Picking up the stretch from the interoceptors or the viscera receptors, picking up the stretch of a blood vessel, of the walls of your gut, your stomach, your intestine. But Ruffini corpuscles can actually pick up, in addition to touch, they can pick up a little, and heavier touch, crude touch, because they're fairly deep. They can also pick up stretch in the skin. So if your skin, for whatever reason, is being stretched, I'm trying to stretch my skin between my thumb and my finger there. If your skin is being stretched, you pick that up with Ruffini corpuscles, or whatever that's worth. Hair root plexus is picking up touch. Pacinian are going to pick up pressure, deep, crude pressure, heavy pressure, and fast vibration, deep. Tendon organs, not a problem all there, totally proprioceptive, and free nerve endings. Free nerve endings. So take a look at what we got. Take a look through the list what we have so far. A, we covered. C, we covered. E, we've done. And H, we've done. Therefore, ergo, thus, does it make sense that the free nerve endings are just going to get everything that we haven't got so far? So just give it a B, D, F, G. It's pretty much going to pick up everything that we haven't got so far. So I'm more than happy to give this a B, D, F, G. Picking up itch, picking up cold and hot, and picking up tickle and pain sensations in those free nerve endings. Take a quick snapshot in your brain. Freeze it, get a little snapshot, get a screenshot because I'm erasing ink, and we're going to go back over on slow and fast adapting. And Merkel's tactile discs are slow adapting. You want to be able to pick up fine discriminative touch constantly. Remember, slow adapting means that it doesn't shut off. It just continually says, hey, something going on. Hey, something going on. Hey, something going on. Continually sending signals to the CNS. And Merkel's tactile discs with that fine light pressure or that fine light touch are constantly telling you that there's something there. Joint kinesthetic receptors. Again, I'm gonna jump right ahead. I'm gonna call muscle spindles very slow adapting with an I. I gotta put an S. I didn't mean S there. I meant an I for Merkel's. I meant an I for Merkel's, right? I for slow. Put an S. And I'm gonna put an I for slow adapting for tendon organs too. The muscle spindles and the tendon organs are constantly monitoring position in space, and there's no reason for them ever to shut up. Again, when is it a bad idea to know where you are in space? It's always a good idea. That's how you avoid massive traumatic injury, by knowing where your body is in space. So muscle spindles and tendon organs are slow. Hey, here's my elbow. Here's my elbow. Still here. Still here. Still here. I still know where my elbow is, right? I still have that muscle spindles and tendon organs. I know the stretch of my biceps brachii. I know that constantly, okay? Why did joint kinesthetic receptors adapt more quickly? Because they're about acceleration and deceleration, right? And acceleration and deceleration don't last very long. So they just kind of picked up quickly and ignored, waiting for the next acceleration or deceleration from the joints. for whatever that's worth. Meissner's corpuscles that are very superficial, they adapt quickly. They sort of tune out. They let Merkel's hang on and continue to send messages. Meissner's tend to tune out a little more quickly. Okay, light touch, light pressure. Let's tune that out and just kind of move on. Slow vibration, no big deal, right? We'll kind of tune that out and adapt pretty quickly to that. Ruffini corpuscles are going to adapt slowly. Okay, stretch of skin is pretty important. And that's a big one. Touch too, but stretch of skin for Ruffinis. You kind of want to know if your skin's being stretched. That's not something you want to sort of tune out, right? Like, oh, my skin's being stretched. Like, that's kind of a relevant piece of information that might change my approach, right? I don't want to tune that out. Okay, hair root plexus, pretty fast adapting, just kind of like tickling those hairs. Again, sort of ruffle the hairs on your integument, the pili on your integumentary system on a hairy area, and then keep your fingers there. They are contacting the hairs, they are moving it, but if you could hold super still, can you just picture in your mind not really feeling that anymore, or that sensation really dimming? That's what fast adapters do. They really just dim out that sensation, like, hey, this isn't important anymore. Pacinian, again, fast, the deep ones that are handling pressure and fast vibration. You kind of tune them out again. Push sort of smoothly with a little bit of pressure, significant pressure on your integumentary system. After a bit, you can start concentrating on other things because that's not really being registered all that strong. And then free nerve endings are actually not going to be either slow or fast, and it really kind of depends on the free nerve ending and what the application is. So feel free to be a little generic with that and just kind of call it slow or fast. It varies. All right. Thank you for sticking with this. Kind of really sums up all of sensation. Now we're going to move into the pathways and how we get all this to the cerebral cortex through our beautiful DCML and our ALS and our trigeminal thalamic pathway and even our spinocerebellar going into the cerebellum. That's what we'll hammer out in the next video. So come join me there, and thanks for watching this one and sticking with it. I really appreciate it.